Good afternoon and good evening, everybody. My name is Sarah DeFeo, and I'm the Director of Scientific Affairs here at Ovarian Cancer Research Fund. I want to thank you all for joining us today as we wrap up our webinar series for 2013. Tonight's topic is exploring the origins of ovarian cancer. Our entire webinar series has been supported by Genentech, and I want to thank them for their generosity in making this possible. OCRF is the largest and oldest charity in the U.S. funding ovarian cancer research. Our mission is to fund scientific research that leads to more effective identification, treatment, and ultimately a cure for ovarian cancer. Since we made our first grant in 1998, we've awarded over $50 million in research grants. Now, before we get started, I want to take care of just a few housekeeping issues. All listeners will be in listen-only mode throughout the presentation, and we will have some time for questions after the presentation. So if you think of a question as you're listening, please type it into the Q&A box that you can see on the right-hand side of your screen. Nobody else will be able to see the questions that you ask, and you won't be able to see anybody else's questions, but I'll see them all, and we'll select some to ask to our presenter at the end of the presentation. And there's no need to wait until the presentation is over to submit your questions. You can just type them in as we go along. I do ask that you try to keep your questions as general as possible, so that the answers will be useful to everybody who is listening. So we're very fortunate to have, to have Dr. Ronnie Drapkin with us today as our presenter. Dr. Drapkin is an assistant professor at Harvard Medical School, an associate pathologist at Brigham and Women's Hospital, and a principal investigator at Dana-Farber Cancer Institute. Dr. Drapkin received his bachelor's degree in biochemistry from Brandeis University and his MD and PhD from Robert Wood Johnson Medical School. He completed his residency and postdoctoral fellowship at Dana-Farber and joined the faculty at Harvard in 2006. Many of his research interests, among his many research interests, is developing a comprehensive understanding of cancer pathogenesis with a special focus on ovarian cancer, which is what we'll hear about tonight. And finally, he's also a member of the OCRF Scientific Advisory Committee and a former grantee. So, Dr. Drapkin, we will pass the mic to you, and you can start your presentation.
Great. Thank you very much, Sarah. I hope everyone can hear me. I am transitioning now. Good. Great. So I want to thank everyone for taking the time to join the webinar this evening and hear about some of the ongoing work in the field of ovarian cancer research. As Sarah mentioned, a lot of the work being done in the field right now couldn't be done without the support of OCRF and the various institutions and investigators that they support. And I was lucky in the past to be one of those. And a lot of the work that started in my lab over the last 5-6 years was, in fact, due to the support from OCRF. So I'm really grateful to them as well. So let me begin with a little bit of history about women's cancers. Many may be familiar with the fact that the term cancer was originally coined by Hippocrates because he described what these tumors look like crab-like structures, and hence came the term cancer. But really, the oldest documented cases of cancers can be found as far back as ancient Egypt, where details of some of the earliest cancers, in fact, some of the earliest cancers were all women's cancers, were describing breast cancers in papyri that were found many years back, described not only the appearance of these tumors, but also some of the surgical procedures that were even developed back then to deal with them, and also the pharmacological and medicinal treatments that were described. In addition to breast cancers, cancers of the uterus and other female organs, including the ovary, are described in these ancient texts, and also the treatment and application of various different medicinal herbs that were described back then, with the hope that they would make an impact on outcome. Now, this more complicated slide is actually quite simple because really it shows us how ovary departs from all the other women's cancers in terms of how it tends to present. So if you can see my arrow, we're looking at the bottom here of localized cancers, regionally disseminated tumors, and distantly disseminated tumors. And what we tend to see in ovary versus the female breast versus the uterus versus the cervix is that ovary tends to typically be presenting with distant disease, unlike breast cancer, which is often found more limited to the organ itself. Same with uterine cancer and same with cervical cancer. Ovarian cancer is oftentimes found a distant disease when we look at staging of patients. So why is that? So let's think about a little bit of the background of ovarian cancer. Worldwide, there's about 215,000 new cases diagnosed every year. Unfortunately, the vast majority, up to 80% of these women, are diagnosed with late-stage disease, as I just mentioned, and that's associated with a poor 5-year survival of less than 30%, a statistic that really hasn't changed over the past few decades. This makes ovarian cancer the most lethal gynecologic malignancy, and part of the reason why it's so challenging is because we have no tools for early detection, and that in turn limits our understanding of what's going on at the molecular level of these tumors. So why is that? Why is ovarian cancer so difficult to detect early, and why does it present with distant metastases? So I think a lot of it can be explained by anatomy. If you look at the panel on the left here, this is a skeletal representation of the female pelvis. And the female pelvis forms essentially a bowl in which these female reproductive organs lie. And so tumors arising in the ovary or anywhere else within the female reproductive system have to get rather large before they creep out of this bowl and create some symptoms that we can identify as being abnormal. And so a lot of times these tumors can become very, very large before we detect them, before you even have symptoms, and that's unfortunately why most of these tumors are diagnosed at late stage. Now, complicating this is the fact that ovarian cancer, very much like breast cancer, isn't a single kind of cancer. So what this illustration is trying to demonstrate is that if this is the uterus here in the middle, the uterus here in the middle, the fallopian tubes, the ends of the fallopian tube we call the fimbria, and the ovary itself, tumors that arise within this region, within the ovarian tubal region, can look like many different kinds of ovarian cancer. So we have isolated or highlighted in the bottom here the serous carcinoma, the serous ovarian tumor, the endometrioid ovarian tumor, the mucinous and clear cell tumors. And it's actually becoming more and more important that when we talk about ovarian cancer, we don't talk about it as one entity, but we actually make effort to really try to understand what type of ovarian cancer we are dealing with. Not only from the perspective that they look different, but we all know that now these all present different clinically. They have a different clinical course. They respond to chemotherapy differently. And so it's very important that we start thinking about applying all these emerging technologies that we all hear about in the news to these subtypes of ovarian cancer, not sort of ovarian cancer as an aggregate. Now, in an attempt to do this, about 10 years ago, there was a group from Johns Hopkins led by Bob Kerman and Yimeng Shi who tried to figure out how can we categorize, how can we classify these subtypes into groups that are amenable to molecular intervention or personalized therapy. And what they ended up coming up with is what they called the two-pathway hypothesis, where you have type 1 tumors and type 2 tumors. And type 1 tumors, they said, were all the low-grade, slow-growing tumors. And by low-grade, I mean the nuclei of these tumors don't look particularly aggressive. They're usually somewhat blander. They're not that aggressive-looking. And these slow-growing, low-grade tumors had mutations in many cancer genes, and the list of these mutations continues to grow as we learn more and more about the genetics of this disease. Now, importantly, many of these low-grade tumors were associated with benign precursors in the ovary. So just one example of that is endometriosis, a very common condition in women. Endometriosis is associated with an increased risk of developing endometrioid and clear cell carcinomas. Now, this is in contrast to type 2 tumors. The type 2 tumors are high grade, so very different than the low grade type 1 tumors. They're clinically very aggressive. This is the tumor that we tend to be thinking about when we say ovarian cancer. We're thinking about this particular tumor. It has the mutations in the p53 tumor suppressor gene, which, as you'll see a little bit later, is quite important. And more importantly, perhaps, is that this is the kind of tumor you get when you have mutations in the BRCA1 or BRCA2 genes, which are the genes that are mutated in hereditary forms of this disease. And until quite recently, over the last 5-6 years, we really didn't have a precursor that we could point to. We didn't know where these tumors were actually coming from. We were assuming they were coming from the ovary because the convention was to call these ovarian tumors at the time of surgery when you saw a big ovarian mass, a big tumor mass near the ovary. We called it an ovarian tumor, but that was really the only reason that we actually called them ovarian to begin with. We hadn't been able to find a precursor in any of these. So why do we want to find a precursor, right? Imagine that you're trying to prevent forest fires, right? Well, the way to prevent forest fires is to prevent that match from ever being lit, right? From ever kindling a small fire that can become a much bigger problem. And so if we can find where those matches are, we can put them out. We can make sure that they never become a cancer. That's the importance of finding precursors. And perhaps the best example of this is the Pap smear that was developed in the 1940s by Papa Nicolau. This is a test that gets done to all women where we can sample the cervix and look at the cells in the cervix and look for these precursor lesions, because if we can find them, we can actually act on that by knowing exactly what kind of precursor it is. Sometimes we perform surgical operations to remove the tissue at risk, or sometimes we can actually wait and watch if it looks to be a low-grade or less aggressive type of precursor. But more importantly than anything is the fact that we know where to look for these precursors, and we can look for them on a regular basis. So, where is the precursor to ovarian cancer? Where is, in these question marks that we have highlighted here, where are these precursors, the equivalent of the match that leads to the smoldering fire that ultimately, unfortunately, leads to the forest fire that ravages all, right? Where is that coming from? Well, there was one clue that enabled some of this work to start being elucidated about 12 years ago, 13 years ago, by a number of different groups. And that's the fact, as I mentioned before, that women who have mutations in the BRCA1 or BRCA2 genes have an increased risk of developing ovarian cancer. Right, 5 to 10% of all ovarian cancers are associated with these familial predispositions. These are the ones that carry these mutations. And the vast majority of these tumors, as I mentioned, are the serous carcinomas, these high-grade type 2 tumors. So this afforded an opportunity because we know that women who have these mutations can have prophylactic surgery to remove their fallopian tube and ovary after childbearing years. So by examining these specimens from these women who are thought to not have cancer at the time because they have no symptomatology and no evidence of it, but are removing the tissue at risk to avoid getting those cancers, we could look at those tissues and ask, where is that early precursor? And that's exactly what a lot of groups over the last 10, 12 years have been doing, looking for where are these early cancers. Again, where is that match that we want to very desperately identify? So this table is not meant to be comprehensive at all, and really all it's sort of highlighting is some of the studies that have been done over the last decade or so where they've looked at these prophylactic specimens and asked where is the cancer. And the whole point of this is simply to show that most of the time when they did find early cancers, it was involving the fallopian tube rather than the ovary. And in fact, in one study, they found that most of these early cancers, in fact, the early cancers they found were all in the distal end, the very tips of the fallopian tube that are brushing up against the ovary, what we call the fimbriated end of the fallopian tube. And this was paradoxical because we were looking for early ovarian cancers, and yet in the cases that we could find early cancers, they weren't in the ovary, they're in the fallopian tube. So at this point, one of my former mentors, Christopher Crum at the Brigham and Women's Hospital right next door, decided to look at this a little bit more carefully. And so he developed what's called the Fim Protocol. It really means sectioning and extensively examining the fimbriated end of the fallopian tube. And so what you're looking at this figure here is the fallopian tube, when taken out of a patient, we cut off the fimbriated end of it and create cross-sections through it to really expose as much surface area that we can examine as possible. And this is what one of these sections looks like under the microscope at low power. And the question was, if any of these cancers are really coming from the fallopian tube, and if we think they may be concentrated in the very fimbriated end of the tube, the part closest to the ovary, what do we see when we systematically use this type of protocol to look at all these prophylactic specimens? Now, in order to appreciate what this looks like, you're going to get a little primer in histopathology. So what does normal fallopian tube look like? Because you really can't appreciate what abnormal looks like unless you know what normal looks like. So here's an example of what normal fallopian tube epithelium looks like. You have what we call these ciliated cells. They have all these hair-like projections protruding from the top of the cells. We call these cilia. And you have secretory cells that don't have these hair-like projections, and we call these secretory cells. So you have ciliated cells next to secretory cells, and they form nice lines and rows of cells of these darker shapes of the nuclei of these cells, right? And you can see them on this side as well. So that's what normal fallopian tube epithelium look like. Cancers of the fallopian tube, or early cancers, what we call tumors that are still within the epithelium, look, as you can appreciate here, like they're all piled up. They're no longer organized in the same way. They have mitotic figures. They're more proliferative. Here's an example of a cell undergoing mitosis here. The chromosomes are lined up at this, what we call the metaphase plate. So they're more proliferative. They're disorganized. The nuclei are bigger than normal. And importantly, there are no ciliated cells in these cancers. It's all made up of those secretory cells that don't have those little hair-like projections. So what did we find? Here's sort of the punchline. We found that many early, quote-unquote, ovarian cancers actually begin in the fallopian tube, and that almost all of these early lesions involve the fimbriated end of the fallopian tube, just the very distal end of the fallopian tube nearest the ovary, not in any other part of the fallopian tube, but we can get into why that might be a little bit later on. So we think that we were able to identify, by using this protocol, the fact that many, but not all, of these early ovarian cancers are beginning in the fallopian tube, something that hadn't been appreciated before. Now, what does this actually look like? So if we look at, under the microscope, we look at normal fallopian tube. Again, you see the small arrow pointing to the ciliated cells and the big arrow pointing to the secretory cells. And what we found in these cases of women who had these BRCA mutations but had no disease was a number of what we call precursor lesions. The first was what we call the P53 signature. And this is made up of cells that look like normal secretory cells, but if you stain for the expression of a protein, as I mentioned, P53 is an important tumor suppressor in all cancers, but particularly important in ovarian cancer. We see that this protein is present here by the measure of brown staining in the nuclei of these cells, and it's not normally there in normal fallopian tube epithelium, so it's only in these signatures. We also looked for DNA damage, and this is a special marker that stains for DNA damage in the cells, and we could find that in these cells, not only did we find stable P53, but also DNA damage. And if we look at this marker called MIB1, which measures how quickly these cells are growing, we found that they're not really growing. They're no more proliferative, we call, than the normal epithelium. But they clearly had something wrong with them because they were staining for DNA damage and they were stabilizing this protein called P53. And in those rare cases, the 5 to 15% of time where we find early cancers in these prophylactic specimens, we found our friend the Tubal intraepithelial carcinoma. Again, these look like cancers. These cells are cancerous cells. They also retain expression of this P53 protein. They have that DNA damage, but now they've acquired a proliferative capacity. Right? The brown staining down here shows that these cells are now growing and they're proliferating faster than their normal counterpart over here. And when we look at the actual ovarian tumor that we see clinically, the tumor shares many of the features that we find in these early fallopian tube precursors, right? They look like cancer, they express P53, they have DNA damage, and they're proliferative. So by studying these prophylactic specimens from women at risk, because they had obviously mutations in BRCA1 or 2, we were finding some of these interesting precursor lesions in the fallopian tube that we hadn't been able to find in any sort of reproducible way in the ovary before. So, can we start filling in some of what's happening in this progression from the match being lit to the smoldering fire to the wildfire? And I think we can. We've identified precancerous lesions we call the P53 signature, the early cancers we would call the Tubal intraepithelial carcinoma, and then obviously the clinically relevant ovarian cancer that we find at the time of clinical diagnosis. So I think now we've been sort of able to fill in some of what we hadn't known in the past, that many of these tumors begin in the fallopian tube, and the cells themselves at some point leave the fallopian tube and seed the ovary, and that's where most of the tumor likes to grow. So the ovary still plays a very important part in the development of these cancers because it sort of is the fertile ground for these early cancers to grow into these large, clinically problematic tumors. So if we put this all together, we can come up with a model where we think that the type 2 tumors, these high-grade tumors, begin in the distal end of the fallopian tube, and the low-grade, or type 1 tumors that I described, begin somewhere within the substance of the ovary itself. So that ovarian cancers are sort of a hybrid. Some begin within the ovary, and some actually don't begin in the ovary itself, but home to the ovary and grow in the ovary, but actually have their genesis, their beginnings in the very distal end of the fallopian tube. So what are some of the take-home messages? I think some of that, it could be that the high-grade serous ovarian tumors are the most common type of ovarian cancer that we know of. It's the most aggressive type as well. Surprisingly, the precursors of these high-grade serous tumors exist in the fallopian tube and not in the ovary. And that this protocol that was developed, the Fim Protocol, is an important clinical tool for assessing these prophylactic specimens from women who are at high risk of developing these cancers. But like with any type of study, and this again is a study that's evolved over the course of a number of years, there are always some nagging questions. Some of them include the following. do all high-grade serous carcinomas come from the fallopian tube? I think that the data that exists right now says we don't know. I can certainly tell you that not 100% of these tumors come from the fallopian tube. The best data out there probably suggests that 60 to 65% of these tumors can be linked to early precursor lesions in the fallopian tube, but not 100%. Another question is, how do we treat patients with these early lesions, or should we treat patients with these early lesions? And I can tell you that early on we really didn't know what to do. I think at this point, if these lesions are still contained within the fallopian tube, what we call intraepithelial, we tend not to treat because we feel like we've gotten it all and it hasn't spread beyond the fallopian tube itself. Another question is, what are the implications for early detection, right? I talked about one of the reasons this cancer is so problematic is that we don't have tools for detecting it early. And part of that is the anatomy of the female pelvis, right? We talked about the bowl and how these tumors are sort of sitting in this bowl, and until they grow beyond the confines of this pelvic bowl, it doesn't create symptoms. So what are the implications for early detection? A lot of the effort was targeting the ovary, looking for clues in the ovary. Maybe we should redirecting our scope, so to speak, to the fallopian tube and looking for opportunities for early detection there. And then maybe the most significant question that's in the field right now is whether there are implications for prophylactic surgery. And what do I mean by that? So as I told you, the folks that the women at high risk of getting these cancers are the ones that carry the BRCA1 and 2 mutation. And after childbearing, many of them are offered the opportunity to remove the tissue at risk. And what does that mean? Well, we used to take out the fallopian tube and the ovary, and that would put women into a surgical menopause. Basically, you would be in menopause because your ovaries are gone at a very young age, much younger than you would normally enter menopause. And the question is, if these tumors really arise in the fallopian tube, could we spare the women undergoing this procedure the early surgical menopause by just taking out the fallopian tube, thereby hopefully reducing the vast majority of the risk, and perhaps going back secondarily after they've had their natural menopause and take out the remaining ovaries to make sure you remove all. And I think there's a lot of conversation around that. I'm not sure we have an answer to that yet. We certainly can entertain more specific questions about it. And then obviously the other questions are your questions, and I'd be glad to entertain any that you might have. And finally, I just want to thank some of the folks and give credit to some of the folks in my lab. Obviously, a lot of the work we do is not done in a vacuum. We're in a day of molecular medicine and personalized medicine where we need to collaborate with each other. And so there's the folks in my group and my collaborators at the Dana-Farber and at the Brigham, especially Christopher Crum and Michelle Hirsch and others who have really helped in not only understanding this observation, more importantly, providing some clinical insight as to why it's so important, collaborators at Mass General Hospital and other places that have really made it fun to work together. And I think I will stop there and entertain any questions you may have. Thank you.
Oh, sorry, you guys couldn't hear me. I had my phone muted. Thank you, Ronnie.
Sure.
I just wanted to remind you all that you can submit questions via the Q&A chat screen on the right-hand side of your screen. So just to start with a general question, so what about the theory that ovarian cancer starts on the surface of the ovary, serous ovarian cancer starting on the surface of the ovary? Is that theory no longer relevant? Are people still doing research? Can you just kind of talk a little bit about where we are with that?
Sure. Well, again, I think that the available data does not at all dismiss the work that's been done on the ovarian surface epithelium. In fact, the reason for thinking that the ovarian surface epithelium was relevant or potentially a cell of origin there, it's because it's really the only epithelium in the ovary. So if these are truly ovarian tumors, then it has to have come from some epithelial precursor, and that was really the only epithelium that we can identify as sort of the normal epithelium there. So, you know, whether it's relevant or not, I think that, well, personally, I think it still is. Like I said, I don't think we can discount the observation that we can't identify 100% of these serous tumors arising from the fallopian tube, and there must be a reason for that. So either there are other sites that it can arise from, like the ovarian surface epithelium, or we haven't been able to, for reasons that are maybe technical, have not been able to identify them all in the tube. But my gut says that it's probably still related and still relevant in that there may be multiple sites of emergence of these tumors, including the ovarian surface epithelium, but that probably the most prominent one is the fallopian tube.
Okay. Got it. Here's a question. As the study covers, the study you presented covers patients with BRCA mutations, which, as we know, is a small portion of the number of cases of ovarian cancer. Can you comment on how this might apply to non-BRCA mutation carriers?
Sure. So I'm going to actually take advantage of the fact that I put some additional slides in my Rolodex. Do I have control of this?
Yes, you do.
I do. So let me just see. Here we go. Great.
Yep.
Okay, so let's go to this. Okay, so I'm going to go back to the slide I showed you before and sort of just break it down a little bit, right? So one of the things I said is the original studies were all done on samples from women at risk, as you mentioned, right? The BRCA1 and BRCA2 mutation carriers, right? And what we found is, in the first two panels that I'm showing you here, is sort of what normal looks like and what these P53 signatures look like, okay? Now, the important part was that when we went back and looked at women who didn't have mutations in BRCA1 or BRCA2, we also found that they had these P53 signatures. In fact, 30 to 40% of all women, regardless of whether they have a mutation in the BRCA genes, have a P53 signature at any given time. And that's simply what we found by examining lots of samples from women who were having their tubes and ovaries or in uteri removed for reasons unrelated to cancer. One example is fibroids. And we would look at their fallopian tubes, and we would find these P53 signatures, right? So that says to me that the emergence of these really early precursors, these really early structures, if you will, hasn't anything to do with genetic risk, but it's physiology. Now, what would that be? So there's some really old literature, 1971, to those that think that that's really old, that says, and this is a hypothesis that was put forth by Fathala, who said that the risk of ovarian cancer is related to how often you ovulate. And he specifically meant in cases where you ovulate without interruption, what he called incessant ovulation. The more incessant ovulation you have, the higher the risk of ovarian cancer. That basically meant women who never had children, who didn't take birth control pills, who didn't breastfeed, had a higher risk of developing ovarian cancer. It also meant women who had early menarche and late menopause, so a long reproductive life, was also a risk for ovarian cancer. And he's argued that the reason that is because the more ovulations you have, the more eruptions through the ovarian surface as each follicle is released every month that occurs, the more injury there is to those cells overlying the surface epithelium of the ovary, and those cells, the more you injure them, the more likely they are to ultimately become a cancer, right? So what we interpret, now we take that hypothesis that's about 40 years old, and we say, you know what, that actually makes some sense. But it may not necessarily just be the ovarian surface that's affected. It may be the fallopian tube fimbria, which is draped right over the ovary to catch that egg every month, that's impacted by the ovulation that occurs and is also injured. And so I think that this injury that occurs every month is what leads sometimes, a third of the time, to these P53 signatures to develop, right? And it's all physiology. It's all related to ovulation. In those women who have the BRCA mutation, then they're more likely to traverse this transition from P53 signature to tubal intraepithelial carcinoma, an actual cancer, an actual pre-invasive cancer. So the genetics comes into play after the fact, right? Ovulation is normal, it's physiological, but it causes a little bit of damage. The genetics enables that little bit of damage to become a problem in some women who carry these mutations. And I think that's where sort of the ones with mutations suffer, and those that don't usually stop here and never progress to the cancer. I hope that answered the question.
Thank you, Ronnie. We got lots of questions coming in here. Would ovarian cancer be treated any differently if it started in the fallopian tube?
That's a very good question. Keeping in mind that the treatment for ovarian cancer hasn't really changed in a really long time. So admittedly, we have sort of lagged behind the targeted therapy, personalized therapy approach that we have seen evolve and emerge so strongly in breast and lung cancer. Right now, we treat tumors that come from the fallopian tube that are called serous tubal cancers, or ovarian serous cancers, or primary peritoneal cancers the same. The therapy is based on giving a derivative of platinum, carbotaxol, and taxane, and that's sort of been the standard of care since the late 1990s, and all of them get treated the same. I think as we learn more about the genetics and the genomics of these cancers, the hope is that we're going to be able to get to a point where we can triangulate on sort of key molecular changes that are amenable to therapy, and we can start developing some of those targeted agents. But at the moment, we really haven't sort of gotten to that point in any sort of standard way.
Okay, thank you. I want to pop back to the previous question and answer, and someone is asking, if P53 signatures can appear as a result of common physiological changes, do you think that there could be a large risk of overdiagnosis of ovarian cancer / fallopian tube cancer, as we sometimes see with prostate cancer?
That's a very good question. We do have to be careful about overdiagnosing. This is a concept and a term that we've been discussing actually among ourselves over the last few months as to, because there has been some literature about the problem of overdiagnosis, was whether we should come up with nomenclature that more or less confers and tells the clinicians whether there's something to worry about or not. So I'll give you an example. We don't report P53 signatures in our pathology reports if we see them, because we don't believe these to actually be cancers. They don't look like cancer cells under the microscope. In fact, if we don't stain for these markers, P53 and DNA damage, we wouldn't be able to see them. So by definition, they're not cancers. They don't look like cancer. They don't behave like cancer. They don't proliferate like cancer. So we don't mention them in our reports. The only thing we mention in our reports is if we see these tubal intraepithelial carcinomas, because those are actually cancers. They're actually carcinomas. They have all the cytologic, all the cellular features of a cancer, and they proliferate. So they have the potential to leave the tube potentially, or invade the basement membrane and gain access to sort of other parts of the body, sort of to get out of the barn, as we oftentimes say. And so we do report that, even though we don't necessarily know that each tubal intraepithelial carcinoma will behave the same. And so one thought is maybe we should call these tubal intraepithelial neoplasms and not necessarily call them cancer, because we don't know exactly that they all behave the same way. And until we know a little bit more about it, maybe we should kind of pull back the reins a bit in terms of what is likely to happen to someone who's diagnosed as such. So I think it's sort of, the verdict is still out. I think we need to figure out exactly what the nomenclature ought to be, and we need to study these a little bit more to know what the propensity of any one of these lesions to progress is. And I don't think we know enough at this point.
Okay. We have a few different questions coming in about primary peritoneal cancer.
Yep.
Can you comment at all about origins of ovarian, or origins of primary peritoneal?
Sure. So that's a great question, sort of related to the distinction between fallopian tube and ovary. So if you think of these type 2 tumors, as I mentioned before, these high-grade tumors, right? The serous carcinoma is the sort of poster child of that category of tumor. And serous carcinomas are always traditionally thought to come from any one of 3 places, right? It's either the fallopian tube, which was thought to be extremely rare, the ovary, which is the prominent serous type of tumor, and then primary peritoneal cancers, which seem to occur and arise and look exactly like every other serous tumor coming from the tube or the ovary, but seem to show no involvement of the ovarian surface at the time of surgery, and the tubes seem to be normal. And so we didn't have a place to sort of pinpoint to say, it started in the ovary and then spread to the peritoneum. No, in this case, it was tumor in the peritoneum and the abdomen, but the ovaries looked totally normal. So hence primary peritoneal carcinoma, because it presumably emerged from the mesothelial cells. So the same cells that cover the surface of the ovary, we call these mesothelial cells. They also line all the organs in the abdomen. And so the thought was that somehow these cells would change in a way that they would look like serous tumors and emerge from there. And that's kind of how we used to think about it. And part of the reason that was is because there would be women, for instance, who would be offered these prophylactic surgeries. They would have their tubes and ovaries removed, and then years later develop a serous carcinoma, and you'd be like, well, how's that possible? We took out their tubes and ovary. And so it was thought that there was still some residual risk of developing these primary tumors in the peritoneum. Now, what we don't know, and what we're starting to find out now, is whether in those cases where they developed cancers years later, was there an early cancer in the fallopian tube that had already shed microscopically to the peritoneum? We took out the tube and ovary, but those cells had already sort of left the barn and became a cancer later. And that's one of the things that needs to be addressed and studied more carefully. Another reason why it's very important to do washes in the abdomen at the time of these prophylactic surgeries, what we call cytology or peritoneal washes, to make sure that we sort of sample as best as possible any free-floating cells that may already be outside the confines of the tube at the time of surgery, because if we see them in the wash, then we already know they left the fallopian tube and there's a potential risk for developing aggressive disease later.
Mm-hmm. Okay, thank you. Let's see. Are there any implications for this research for treatment of existing ovarian cancer versus detection/prevention?
So this type of work, I sort of think of it as pathogenesis, right? Trying to understand how these tumors arise. I think it's useful for many things. I mean, I think we can think about redefining not only how we try to find these tumors and where we look for these tumors, but our approaches to early detection, and I think our approaches to clinical management may be altered by some of these observations as we've touched on. Whether the treatment will change, as I mentioned before, it really hasn't changed for quite some time, will depend, I think, a bit more on the genetic characteristics in studies that have been going on over the last few years, more on those than necessarily these types of studies. And I didn't really touch on that, but it is really quite relevant, because at the same time that we were making these observations in the fallopian tube and where these early cancers are coming from, there was a big national effort called the Cancer Genome Atlas. We shorthanded TCGA for Cancer Genome Atlas. It turns out to be that those are also the 4 letters of the DNA. This was an effort that was funded by the NIH to support many institutions across the country, and the goal of that was to develop this very comprehensive compendium of all the genetic alterations in cancer, right? So what was the landscape of all these cancers, right? And they started with 3 cancers. They started with brain tumors, lung cancers, and ovarian tumors. And they picked those 3 because those 3 are particularly aggressive cancers for which we have no early detection and we have no good therapy. So the idea was, if we understand the genetics of these cancers, could we make an impact on those metrics, on early detection and therapeutics and so on? And so ovarian cancer is one of those 3, and the study for that was published in 2011. And it really defined the genomic landscape of what these tumors look like. It validated some of the things we already knew. It opened up opportunities and new information that we didn't know. And so the hope is that by delving into that now more and trying to find out what are the genes that are altered that are likely driving these cancers, or driving these cancers to be chemo-resistant, or driving these cancers to recur and progress more aggressively, we can target them molecularly, right? So I think that that's the opportunity where changes in therapy may actually have an impact on the treatment of these cancers directly, not so much where they come from. Okay,
next question is, someone said, I noticed that you had RAD50 or RAD50 noted on your slide. Do you have any information on the role that this variant might play in ovarian cancer?
Yeah, great question. So I think the question is sort of getting at, are there any other genes like the BRCA1 or 2 genes that are likely to give hereditary predisposition to developing of these cancers? So people have been looking for a BRCA3 for a long time. In fact, the early studies to try to find these genes was in the mid-90s. Was really sort of combing the genome and looking at all the genes and looking at families who had really strong hereditary patterns of inheritance for these cancers. And that's how BRCA1 and 2 were found. And really, we didn't see another sort of smoking gun that we would call BRCA3. It turns out that BRCA1 and 2 account for a good chunk of all hereditary cancers, but there's definitely a big slice of the hereditary pie that isn't accounted for by these mutations, but seems to be accounted for by a whole collection of other genes, including the RAD50 and RAD51 and PALB2 and BRIP genes. Interestingly, all those genes it's sort of, they're not unrelated. They all cooperate with BRCA1 and BRCA2 for DNA repair. That's their normal function. The normal function of BRCA1 and 2 is to coordinate DNA repair in the genome, to preserve the integrity of your DNA. We're exposed to all sorts of DNA damage all the time, from external X-rays and sunlight to endogenous forms of DNA damage. I mean, normal replication of your DNA and cell division can cause stress to the DNA. And so we have very robust DNA repair mechanisms in our body. And it turns out in cancers, defects in these oftentimes are sort of the key component that leads to the development of the cancers, because they enable mutations to accumulate and provide these tumor cells, these fledgling tumor cells, with a growth advantage. So in fact, there is data, in fact small but not as large family cohorts of hereditary ovarian or breast cancer or both that have mutations in these RAD50 genes and these PALB2 genes. And then the question is going to become, do we know enough about their contribution to what BRCA1 and 2 is doing that we might be able to sort of think about offering not only sequencing for these genes to these folks with hereditary cancer, especially when they go for testing and they're found to be normal for BRCA1 and 2, might they have a mutation in any of these other genes? That right now is not something that's clinically offered. I think you heard earlier this year from Elizabeth Swisher at Washington University in Seattle that she's developed a platform for sequencing all these other DNA repair genes and looking for mutations in these women who have hereditary cancer but have wild-type BRCA1 or 2 genes. I think that's going to be an area that will evolve over the next few years and may actually expand the platform and the portfolio of genes that we can look for in terms of these hereditary cancers.
Okay. A question that just came in, and I'm not familiar with this, so I'm going to ask you. This person says, there are folic acid receptors in ovarian cancer. Could radioactive folic acid be an approach to therapy?
Yep, that's a good question. It's an area that I was just at the European Society of GYN Oncology meeting in Liverpool a few weeks ago. And I'm not an oncologist, I'm a pathologist, but I can tell you what was reported, which was that they are trying to target the folate receptor, and they developed a monoclonal antibody to it. Now, I can't recall if that monoclonal antibody had a cargo, a payload of chemotherapy or not, but the idea was, if we can target these folate receptor expressing cells, could we delete them? Could we cause them to regress? And unfortunately, the data on that was negative. It didn't seem to confer any benefit in terms of tumor regression that I could recall. I don't know that that's been published. It was reported at the meeting. It was somewhat disappointing, but definitely folate receptor is an area that's gotten a lot of attention from the big pharmaceutical companies. It's clearly an interesting, viable target because it's expressed on the surface of these cells, and so it's easier to get at it. And there may be other opportunities to target it, like radiotherapy may be more effective than a monoclonal antibody or carrying a chemotherapeutic payload. And I'm sure in fact, I know of a few companies that are developing a number of folate receptor-based approaches to ovarian cancer. So we'll see how that emerges and evolves over the next year or two.
Okay, we'll start to wrap it here. Just a last question. Going forward, what do you suggest might be the most promising method for early detection of some of these precancerous lesions that you mentioned in your presentation?
Yeah, that's a great question. I think it always ends up coming back. So now we find these lesions in the fallopian tube. Now what are we going to do about it? How are we going to find these earlier? And that's a great question, challenging question. I think that's one that we're all grappling with now, is sort of how to redefine and readjust our approach to looking at this. So I think there's a number of ways you can do it. One way, one approach we're taking in the lab, and others as well, is to ask the question, well, now that we know where these tumors come from, right, if what we're aiming for is a biomarker that we can detect in the blood, then we're really looking to ask, what do these early cancers secrete that they don't normally secrete in the fallopian tube? Or are there any proteins that are secreted by these early cancers that aren't secreted by the fallopian tube? And basically you're saying, is there something in normal fallopian tube that is gained when it becomes a cancer here, right? So does this, what I'm circling here in these tubal intraepithelial cancers, even in these early invasive cancers, do they make something that normal tube doesn't, or do they make more of something that's normally made there? And so one of the things we've been trying to do over the last few years, see if I can show you. So one of the things we've been trying to do is develop these platforms, these model systems that are based on the fallopian tube epithelium as a basis for trying to address some of these questions, right? So for instance, down here, what we call this ex vivo bioanalytical platform, all it simply means is that we've been able to figure out a way to grow normal fallopian tube cells directly from patient samples in the lab in such a way that we can monitor what they normally secrete. And now we can do that with matched normal and tumor. And so we can compare what does the tumor secrete that isn't secreted by the normal, or what are the ratios in terms of things that are secreted by both. So that's one way you could do it, right, is looking to see what normal versus tumor secretes and try to make tools and detection metrics to look for that particular protein or set of proteins. The other is an imaging thing, right? Can we figure out a way to image these early lesions early so we can know when to go after them and when to surgically remove them? And that's an area it's very interesting, a lot of interest around that. Again, a lot of it will depend on finding something about these early tumors, early lesions that is different than the normal, so that we're actually finding something that's cancerous and not some normal structure. And there's a lot of cool technology next door at MIT and at the Broad Institute that's being developed, nano technology, where we can deploy imaging sensors into the fallopian tube, or antibodies labeled with contrast agents that might be able to detect abnormalities in the fallopian tube that are imageable. Lots of different approaches are being utilized to try to develop that, but I don't think we've gotten to the point where we have something. I think a lot of attention is being put to it, and hopefully we'll see something emerge in the next couple of years.
Okay, thank you so much. So I want to thank Dr. Draper for his presentation and for answering so many questions. And I want to thank everybody for calling in today. This will be the last webinar for 2013, but stay tuned for details on the webinar series that we'll be developing for the new year. Please take a minute to fill out the survey that will appear on your screen at the conclusion of the event. Your feedback is very important to us and helps us come up with ideas for new webinars. And for more information about OCRF, you can always visit our website at www.ocrf.org. So thank you all, and have a great evening.
Thank you, everyone.
Thanks, Ronnie.